So far we have discussed about some facets of fluid mechanics over small scales, and just to get a little bit deviated from that perspective, we would like to now focus on, like, when we are thinking about fluid flow through small passages, how are those small passages fabricated. So how do you make micro channels and micro scale passages which are important for microfluidics based applications. So for that purpose we will have a brief introductory lecture on microfabrication. Please keep in mind that microfabrication is a subject by itself, or micro manufacturing is a subject by itself. Our objective of this course is not to get into the details of microfabrication, but give you some idea that what are the traditional perspectives, what are the current outlooks, and what are the futuristic perspectives of microfabrication. So microfabrication versus macrofabrication, like I mean, because many of you are actually with background of mechanical engineering, you have ideas of classical macro scale manufacturing processes like casting, welding, forming and all these, and now you have to realize that when we are thinking about manufacturing over small scales, you are not directly using those processes, but some of the concepts might be closely associated. So when we are talking about a manufacturing process, we are essentially interested about two types of possibilities. One is called as top down approach and another is called as bottom up approach. Top down approach means shaping of features from bulk systems using tools such as milling, forming, lithography techniques and so on. So basically you have a large system, I mean a system which is of larger size than the final product, and then you shape off some parts from the system to get your final product. So the control is determined by the limitations of the fabrication tools, and but integration into devices and handling is easy, but may lead to wastage of costly components. Bottom up approach involves assembling of the systems to form in more complex structures such as lamination techniques, like for example rapid prototyping, self assembly. So you have small parts of the system and sometimes you assemble those small parts to make the final product, and these small parts may be assembled by sometimes self assembly or self organization. So that is also a possibility, that is because of inherent chemical and biological features associated with such systems. I mean there are tendencies that some atoms may tend, or some ions may tend to combine with some other types of ions, some entities may tend to combine with other types of entities, and that is how a complex shape or topography may be formed. So careful control of chemical reactions which are backgrounds of such systems are necessary for bottom up approach. These are less expensive and these are able to manufacture features that are beyond the capability of top down processes, but you cannot really make very large systems using this. So there are plus and minus points associated with this. So when we say microfabrication, our main goal is micro channel fabrication actually, because that is where our fluid flow is taking place, where the microfluidics is mainly concerned. So micro channel fabrication typically I mean you are bothered about either the classical paradigm where one is thinking of photolithography, soft lithography, we will discuss about this, and micro machining, mechanical micro machining. I will give you some idea of how mechanical micro machining is done. Then micro machining on CD based microfluidic devices, this we have discussed in the previous lecture, and paper based microfluidic devices. So photolithography, pattern transfer through molten wax and toner printing. So these are some of the possibilities. We will start with photolithography. Photolithography is basically printing by light. So what you are basically doing, is a process of transferring an image from a photographic mask to a resultant pattern on a wafer. So basically there is a mask and the mask has a pattern, you transfer the pattern onto a wafer by a suitable method. We will discuss about what is the suitable method, and I mean it is basically transferring a shape from the master pattern to the final object, that is what it is. It requires several things like photoresist, spin coater, mask, mask aligner, developer, baking ovens, etc. These are some common items that are necessary, and photoresists are of different types, and these are sensitive, made sensitive to either ultraviolet, these are chemicals basically, sensitive to ultraviolet light, electron beam, X-rays or ion beams. So based on these you call these as UV lithography, E beam lithography, X-ray lithography, ion beam lithography, depending on what optical source or what energy source you are using. So you can see here that first, so this is an example of photolithography. I will get into a more detailed example, but just to give you an idea, that is sometimes instead of giving theoretical description, if we give a diagrammatic description it becomes more clear that what is happening, that is what we are trying to do. So let us say you have a silicon substrate, so you can see here that there is a silicon substrate. On the top of that we can have a layer of SiO2. We will see later on that why this oxide layer may be necessary, but it is not always a must that you have to use this layer. And then you basically independently you prepare a mask. In the mask you have a certain pattern. So by the name mask it implies that if you have an energy source, some part of the energy source will be masked by that particular mask where there is a pattern. So that black region will mask the fall of the energy source, and then what you do is, so you coat the wafer with oxide layer with a photoresist. So photoresist is a particular chemical which will on which you expose the light through a photomask. So you can see here, say you have, this is the mask, the black region are the patterns. So the black regions are the patterns, so through these the light energy cannot pass. So you are having the light falling on selective portions of the photoresist. Now when the light is falling on selective portion of the photoresist, either the photoresist will be sort of more soluble in the developer when it is developed and go away, or it will be less soluble in the developer and it will not go away. So if you now develop it with a developer, you will see, for example, that here we are talking about a photoresist where the photoresist has become more soluble with the developer and it has gone with the developer. So wherever it was exposed, see this part of the photoresist was not exposed, this part of the photoresist is below this black region, right, it is below this black region, so this was not exposed, but whatever was exposed, when you develop it in a developer, that exposed part is gone. That is a special type of photoresist. We will see what are the different types of photoresist, positive photoresist or negative photoresist. So then that means what you have been successful with is you have now transferred the pattern from the master or from the mask to the substrate, right. So that is what you have been able to achieve. And then what you can do is you can use an etchant to etch the film. So if you are using an etchant to etch the film, so you can see here that this is the structure that you eventually get. So if you have, so you see that this black SiO2 film, So if you use an etchant, then the part which is exposed to the etchant is gone, so this black part is gone. So only the part which is not exposed to the etchant will remain. So you can see a groove like structure has been formed on the silicon substrate. So now we will so we have a broad idea that what is the process. Now what we will do is we will get into a little bit more descriptive details of the mask. So mask is a template that allows selective exposure of a photosensitive surface to the pertinent radiation. Mask making, so making a mask is very important, and mask making consists of two steps. One is the layout of the design and then the pattern transfer to the mask. So typically the layout of the design you do it in a CAD software, like say for example AutoCAD or some kind of CAD software, and then you transfer that pattern to the mask. What are the properties of the mask? The mask must be transparent to optical wavelengths and must have extremely flat defect free surfaces. It is preferable that it has a coefficient of thermal expansion very similar to silicon, right. So it is just like, think of the mold and the pattern in the casting. The pattern is like mask. So I mean if you have an unequal thermal expansion, then that can give rise to defects. So basically, so if you are transferring from the mask to the silicon substrate, and then it is good that if they both of them have same coefficient of thermal expansion, so they respond to temperature in a similar way. Commonly used substrates are silicon wafers, soda lime glass and quartz. Quartz masks have very similar coefficient of thermal expansion to silicon, but are somewhat more expensive than soda lime masks. Any temperature variation may have significant effect on masks with high coefficient of thermal expansion, and a small change in the dimensions of the mask may translate into big change in the position of fine features. Remember, we are thinking of micron size features, so even a small change in temperature can create a significant change as compared to micron size dimensions. So mask making, the layout for each layer is drawn using a CAD software. The patterns of microchannels and chambers are printed on two different masks with aligned marks. The CAD generated patterns are printed on the appropriate substrates. Silicon wafers, soda lime glass, quartz, or even transparency sheets. Transparency sheets are also, I mean many of you might not have seen transparency sheets, because I mean nowadays we use LCD projector for projecting presentations, but if you have seen a little bit old day presentations, I mean there are transparency sheets which are available for making overhead presentations. So transparency sheets are not very expensive sheets, and one can use this for like mask making. Now growing up, I mentioned that on the top of the silicon, like silicon is just an example, because I mean this lithography technique is actually borrowed from the semiconductor industry. It is not a mechanical fabrication, mechanical manufacturing process. It is basically an electronic fabrication, electronic circuit fabrication process, from where we have got this particular process borrowed to make microfluidic devices. So silicon is being one of the very common materials there, and silicon is also commonly used for many microfluidic applications. So we are giving silicon substrate as an example. So why oxide layer is grown on silicon substrates? There are two particular reasons and these reasons, if they are not important for your purpose, you need not use the oxide layer. So one is to make the surface of the silicon hydrophilic, and the second is to protect specific zones of the surface where etching is not possible. So there are different thicknesses of oxide layers that are formed, and typically protocol is that silicon substrate is kept in oxygen furnace at around 1200 degrees centigrade and dry oxygen is passed through around 2 hours. So these figures are not important for you, I mean it is not it is important for practicing engineers, and when you practice in the lab this may be important, but I mean I can assure you that these numbers 1200 degrees centigrade and 2 hours and all those things will not be important so far as your exam related issues are concerned. Depending upon the thickness of SiO2 being obtained, duration of the heating varies. So that is like these are sort of sometimes these are done through simulations, the optimization and all, but most of the times these are actually trial and error based figures. To obtain thickness above 500 nanometers, oxygen with water vapor is passed in addition to dry oxygen for different time interval depending upon the thickness of SiO2 required. So an important note is that development of oxide layer is not a must for making microchannels. It may be required for specific applications like making of electronic device, protecting specific zones for fabrication, etc. So not a must. Now on the SiO2 layer combination, we coat with a photoresist, that is the next level of coating. Before we shine the entire system with a light source which is masked by the mask with certain patterns. So when we do that, so photoresist, how the photoresist is deposited, it is basically deposited by a process called a spin coating. So there is a device called a spin coater which basically deposits a thin layer, thin film of the photoresist on the substrate. The photoresist may be either positive or negative. Negative photoresist becomes less soluble in the developer solution when they are exposed to radiation. So when they are exposed, so this is an example of a negative photoresist. So negative photoresist, when it is exposed, it is less soluble to the developer solution, so it is remaining. On the other hand, positive photoresist becomes more soluble after exposure, so here the photoresist is gone after development. So basically you can see that so far as this groove is concerned, you depending on the positive or the negative photoresist, you get either the positive replica or the negative replica, but the pattern is transferred. Positive photoresist can give higher resolution, typically they are aqueous based solvents and they are less sensitive. Negative photoresist is more sensitive, relatively tolerant to developing conditions, they are having better chemical resistance, and one of the very important points that they are less expensive, and that is why I mean a very common negative photoresist is used in many labs which is called as SU8, very common negative photoresist, and but it gives lower resolution and typically organic based solvents are used. So here I would like to give you a demonstration of fabricating microchannel grooves, and these microchannel grooves I mean these dimensions and everything are based on experiments which have been done in our lab to fabricate micro heat pipes. So in one of our initial lectures I discussed about that what is a micro heat pipe and how can it help in electronic cooling. So I will show you that what are the steps which you do to achieve it. So you have a wafer, so typical dimensions like this is 275 micron, on the top of that SiO2 which is the blue which is 1 micron, photoresist 0.5 micron, then exposure under UV through mask plate. So this is the mask, so you can see the mask is masking the UV light and this is allowing the UV light. So when this UV light is coming, so now you have the cross-linked photoresist after you develop with the developer. So typical developer is HNR120, this is just an example of a developer. So you have the cross-linked photoresist, then etching of the exposed SiO2 using buffer HF. So you see that whatever is the exposed blue colored region, that is the SiO2, that is now gone with etching with a buffer HF. then the photoresist itself is removed. Photoresist, the pink colored material which was there, the cross linked photoresist, that photoresist is removed by immersing inside a solution called as Pirhana solution. So that is removed. Then the silicon which is there, see SiO2 is protecting the silicon at certain places. So wherever the silicon is there, the silicon is etched by KOH solution, which is a 44% solution in deionized water, to obtain V groove channels. And because of the orientation of the natural slip planes and all, you will not get a rectangular shape, but the etching will give rise to this V groove. And typical etching is done at 70 degrees centigrade for 1 hour 30 minutes. These are again like experience based numbers. Then you remove the remaining SiO2 by dipping in buffer HF. So you can see that nice micro grooves are formed. These are like groove shaped microfluidic channels. You can use these as heat pipes. So this is a summary of the process, and I did not get into the details because we have already discussed about the photolithography process. Now a process which is sort of related to the lithography technique but uses soft materials is known as soft lithography. So soft lithography refers to a family of techniques for fabricating or replicating structures using elastomeric stamps, molds and conformable photomasks. It is called as soft because it uses elastomeric materials, most notably PDMS. Soft lithography is generally used to construct features measured on micrometer to nanometer scale. So there are many techniques of soft lithography like micro contact printing, replica molding, micro transfer molding, micro molding and soft assisted micro molding. So these are all soft lithography techniques. I will show you 1 or 2 typical examples of how the soft lithography is done. But by this time you have heard from me about PDMS several times, that PDMS is a material commonly made for microfluidic channels and for many biological applications, and the question is why PDMS? Why do you use PDMS as a very common material? It is a flexible elastomer, it can be used to replicate topography from a master, it can be used as a conformable stamp for patterning onto other substrates, it is good for sealing microfluidic devices, it can be sealed to many materials, it can be spin coated, it is possible to dry etch, it is very well suited for applications in biotechnology, it is well suited for applications in plastic electronics, well suited for applications involving large or non-planar surfaces, and it is a low cost pattern replication process. So the whole idea is that if you have a good mask, then that is a master pattern that may be obtained by a sort of expensive process, then to make structure on PDMS you do not have to use that expensive process, you do not have to do the standard photolithography which requires a very controlled environment called as clean room. We will come to the concept of clean room later. So you can make the soft lithography process in a clean room free environment. Literally it is not clean room free because the mask that you have made requires a clean room, but once you make a mask you can replicate it to make several structures by using the PDMS. So you can have low cost pattern replication. So you can see, so this is a pictorial demonstration. So let us say that your mass is SU8 based structure. So pour PDMS onto the SU8 structures form, heat it, then peel off the PDMS layer. So it is just like casting if you see. So you see the PDMS that is being poured in the figure, then heat it, then peel off the PDMS layer and bond the PDMS mold with a glass, and then you make of plasma treatment and heat again. So simple schematic representation, the previous was a diagram to represent the generic process, but how do we make micro channels using that? So that is what is demonstrated in this figure, which is of particular importance to us for microfluidics based applications. So you have let us say that this is a mask and this is the SI structure on which you have photoresist. So you have developed a master. So this is the master pattern. Okay. Now you have kept the master pattern and then you cast PDMS on the top of that. Okay. Then you remove the elastomer from the master. See, this is the shape that the PDMS has assumed. Okay. Why this is the shape? Because this yellow colored region is there the PDMS is not filled, right. So just imagine that this white colored region is lifted away, right. So if the white colored, it is peeled off. So if this is peeled off, you will get this groove shape and then you seal it against glass after plasma treatment and insert tubing. Okay. So you seal it against glass. So you make a bonding between the PDMS and glass and the grooved region will make microfluidic channels. So this is how we commonly, this is our day to day activity of like our labs, that is what we commonly do, very simple process. Once you are familiar with it, it is a very simple process of making microfluidic channels for biological applications. Clean room concept. Now as I told you that many microfluidic channels are made by the lithography technique, the standard lithography technique, and which requires a very clean environment to be maintained. So this type of environment control is just like the kinds of controls that are necessary for the ventilation or the intensive care units of hospitals. So you require very sophisticated dust free environment, so that you know your intricate dimensions which you intend, they are not disturbed by deposition of dust particles. So a clean room has controlled level of contamination that is specified by number of particles per cubic meter at a specified particle size. So typically ambient air outside in a typical urban environment might contain as many as 35 into how many, 10 to the power 6 particles per cubic meter, and typically like in the busy urban places that are there in India, it might be even more, I do not know, per cubic meter, 0.5 micron and larger in diameter, corresponding to an ISO 9 clean room. So clean rooms are classified according to the number and size of particles permitted per volume of air. Large numbers like class 100 or class 1000 denote the number of particles of size 0.5 micron or larger permitted per cubic foot of air. Okay. So typically so if you are told that it is a class 100 clean room, so that is what it will mean, that you will have maximum number of 100 number of particles of size 0.5 micron or larger permitted per cubic foot of air in that room. That is the definition. So there are several standards like ISO standards and all those things for specifying clean rooms. I have put this in the slides which will be available to you as notes, and so you might read them, and if you are becoming a practicing engineer in this particular field, these will be essentially important. Now we will discuss something about clean room free fabrication. See, clean room, maintenance of clean room is quite expensive. So until and unless you have very large number of products, then it may not be cost effective. Like clean rooms can still give rise to cost effective products for the semiconductor industry, because the large number of consumers that are there. But when you are thinking of microfluidic channels, currently microfluidic channels are not consumed by so many large number of consumers. So maintaining a clean room for making microfluidic devices has been a bottleneck of many research institutes and many educational institutes which may not be able to afford such things. Even if they are able to afford, it is not possible. So I mean many people are trying to simultaneously get jobs done by the same facility. So you may have to wait for long before you get a chance for your channel to fabricate, but you cannot stop your research there. So you must be having an alternative low cost paradigm where you might not be getting that kind of sophistication, but whatever you will be getting will be good enough to sustain your research in the area of microfluidics. So one of the possibilities is like going for mechanical micro manufacturing. So for example, micro milling. When in our lab we started work with like micro channels, this is the method that we first used to make microfluidic channels. So it is an inexpensive fabrication procedure. The width of the micro channel is determined by the width of the milling cutter. So if you have a small width milling cutter, you can have, and these are available, you can make small width micro channels. Conventional micro milling cutter specifications, I mean these are typical to what we have used in our lab, so do not generalize it. Number of teeth 64, outer diameter 30 millimeter, width can vary from 100 to 500 microns. Nowadays we are getting width even less than that. So 100 to 500 microns channels very easily we can make using these, and feed rates can be varied as 12, 15 and 20 millimeter per minute using this particular system that we are having in our lab. Can you say why the feed rate is important? So your surface roughness characteristics is dependent on the feed rate. So feed rate is very important, and both up and down milling processes can be performed. So you can see that like this is the rotation of the cutter and this is the feed direction. If they are opposing each other, then that is up milling, and if they are in the same direction, that is called as down milling. So both up milling and down milling facilities are possible in micro channel. So typically when we are using micro milling, we are using a material which in loose name is called as perspex, or some people call it plexiglass, and it is actually a polymer PMMA, polymethyl methacrylate, PMMA. Just like PDMS, it is also a very common material in making microfluidic channels. It has a density lower than that of glass and is not brittle. It is softer and hence can be scratched more easily than glass. It can be easily formed just by heating to the normal boiling point of water. It transmits more light than glass, and unlike glass it does not filter UV light and it is less expensive. So it has so many great advantages, and you can have, because of its transparency, you can visualize the flow. So when you are doing fluid mechanics, fluid dynamic studies, so you have a say PIV system, so by which you are visualizing the flow. So it is very nice, it is very convenient to visualize the flow because of its transparent system, but it is not as brittle as glass. So typical like how the things will look, this is an integrated micro channel setup with pressure tappings and tubings for pressure driven flow. So you can see that basically there are two blocks of PDMS and PMMA, two blocks of PMMA. On one block one groove is made by micro milling, on another block another groove is made by micro milling, and they are just screwed together. So the two grooves, U-shaped grooves together form a micro channel. Fabrication of CD based microfluidic devices, I will not go into the details of this because we have already discussed it in the previous lecture. So these might be in your mind already. So these are usually fabricated by stacking and pressure bonding of CNC machine polycarbonate discs, and various diameters of milling bits and drill bits are utilized for CNC machining in the microfluidic network. So the CNC machine CD system typically 5 layered structure has 3 polycarbonate discs and 2 pressure sensitive adhesive layers. So these are the 5 layers, and I have shown you in the last class that how these layers are made and how these layers are bonded together. Now paper based microfluidics. So why are we interested about paper based microfluidics? In CD based microfluidics you require I mean there are several advantages that you can make many microchannels active simultaneously by rotating the disc. If there are many channels, all the channels will become active. So you can have multiple tasks being executed by that system of channels. However, it is always important for the developing world that to go for microfluidic substrates which are even less expensive, and the least expensive substrate that we can think of is a paper. So can we make microchannels on paper? So this concept was first introduced by Professor George Whitesides in the Harvard University, that how do you think of making paper based microfluidic channels. I will talk about some innovations today that what we have made in our lab concerning this, and maybe in a later lecture when we talk about the biological applications of this, I will talk about 1 or 2 more innovations. So what kind of paper you can use? Filter paper, nitrocellulose paper, these are commonly used, but you can use any sort of paper, just try it out. I mean it is just a matter of your effort. These are cost effective, miniaturized, one can use this for multiple tasks, and operational technical expertise is not necessary. So even a very unsophisticated or little bit trained person with not very sophisticated level of training can work with this. So basically what you are interested to do an ideal for point of care diagnostics. So what essentially you try to do in the paper based microfluidics? See, paper has a large number of pores. So if you put fluid in a paper, it will flood the entire paper by diffusing through the pores, and paper based diagnostic devices without having microchannels are available in the market. Like if you see for example the pregnancy test kits, I mean you will see that I mean those are like paper based devices. So any paper based device is not a paper based microfluidic device. Paper based microfluidic device is a device where you make microchannels on paper. How do you make microchannels on paper? You basically block the pores in certain transverse directions so that fluid will flow only along the longitudinal direction, that is the direction of your microchannel. So for that we usually are interested for low cost way of making microfluidic channels on paper, typically where no sophisticated infrastructure or clean room facility is necessary. But you can do you can make microchannels on paper by lithography. For example, we will begin with that. You can for example soak the chromatography paper with a photoresist, and then you have a mask which is a design prepared in AutoCAD and printed on transparency sheet using a normal 600 dpi printer. With this you can have a minimum feature size of around 200 micron. So for this you do not require actually a clean room based process, but it's just a lithography process. So it is pre-baked at 110 degrees centigrade for around 10 minutes and exposed to UV light. So you can see that this structure which is there in the transparency, it is transferred to the paper, and then it is post baked at 110 degrees centigrade for around 10 minutes and rinsed in acetone. So you have the cross link photoresist, which is this violet color that you see, and the white colored region is the paper channel. So it is just like the lithography process, but you can make even paper based microfluidic channels in a simple way. So this is a recent innovation that we had made in our lab. So when we have a paper based device, in the next chapter that we will study electrokinetics, that is how by exploiting electrostatics with hydrodynamics we can actuate fluid flow. So if you can now apply electric field, you can make the fluid flowing through the paper based device at a very fast rate, and if the paper based device is handling blood, that means you can diagnose blood very fast. So to do that, you know, you have to apply electric fields, and depositing electrodes on microfluidic systems are usually considered to be expensive processes. So what we did is, instead of using any standard electrode deposition technique, we scratched the paper with pencil, and this is what is shown in this black region, and this graphite in the pencil acted as electrodes, and then you connect it with copper wire leads. So this is now a paper and pencil device. So if you have a low cost microchannel fabrication system on a paper, couple that with the pencil based electrode scratching, then you can have a very low cost diagnostic platform which is ready for use of blood testing. So there are fabrication. Now how do you fabricate microstructures on paper? So you can have physical blocking of pores, that is what one possibility that I had told, or physical deposition of reagent on a paper, chemical modification of the paper, or 2-dimensional shaping or cutting. Accordingly, there are several techniques which are available, like photolithography, PDMS printing, laser treatment, inkjet etching, wax printing, wax screen printing, flexographic printing, plasma treatment, inkjet printing, and so on and so forth. So depending on, so either you block pores or you make new structures on paper. So basically your objective is to direct the liquid flow in a certain direction on the paper. So this is a different way in which you can do it as compared to what we showed in the previous slides. So pattern transfer through molten wax on a paper. So how it is possible? So you have a AutoCAD design that is printed on this PMMA sheet, then you wax it and heat at around 70 degrees centigrade. So at this temperature molten wax solidifies within the groove which is made. So molten wax solidifies when it cools down to room temperature. So this basically melts the wax at 70 degrees centigrade, and when you bring it to the room temperature, it will solidify, and it will solidify in the groove, but there will be some excess wax. So the excess wax is removed, that is spread over the sheet, and the PMMA sheet is clamped with paper and heated at 70 degrees centigrade for 10 to 30 seconds. So then this wherever this wax is there, that is now, when it is clamped with paper, then there is a transfer of molten wax on paper by fixing the paper with the PMMA sheet. So from the PMMA sheet the wax will be transferred, the wax pattern will be transferred to the paper sheet if you clamp them tightly. So this is basically pattern transfer. There is another very low cost fabrication of paper based devices. I mean, these typical low cost fabrication processes we follow in our lab, so that is why I am discussing about this. So you make toner printing through inkjet printer. So what you do is that printing is done on each side of the paper using inkjet printer. A typical commercial inkjet printer name is given, but it is not a must that you use it. Once the print was done, the paper was heated on a hot plate at 200 degrees centigrade for 4 to 5 minutes, so that toner particles can properly embed within the pores of the matrix. So this is how, so basically you block the pores by embedding toner particles. So this black region is the toner particle. So although you have pores, but what you do is that by embedding toner particles in the transverse direction, you do not allow the fluid to flow. So fluid will flow only in the longitudinal direction, but you have to do it on both sides of the paper because the pores are 3-dimensional structures. So if you do it in one side, then you can see fluid is leaking, so that you cannot do. So using this you can fabricate complex structures on paper. So like you can use paper as a mixing device to make microfluidic mixing, study reactions and so on. Now what next? See, we have discussed about microfluidic platforms, starting from microfluidic channel fabrication platforms, starting from high cost paradigm to low cost paradigm, ultra low cost paradigm, whatever. But what are the new research issues? So you have to think of that when we are making micro channels, many times our objective is to study the biological phenomena inside human bodies. For example, how blood is transmitted through a complex topographically complex arrangement of network channels. So that is one thing. Sometimes we are interested to study drug delivery. For example, in some cancer treatments it is necessary that you basically do a arterial chemoembolization. That means what you do is that you pass a particular medicine, you load a particular type of bead with a particular medicine and pass that medicine through small passages of arterial and venal networks, and then what you basically do is that this medicine will be targeted to the tumor cells. So what they will do, they will serve two purposes. One purpose is that they will block the supply of oxygen to the tumor cells. So that will give rise to ischemia and cell death, but that does not mean that the cell cannot do DNA replication. So for that you have to destroy the cell chemically. So what for that you basically supply drugs on the tumor cells. So you actually achieve two purposes. One is you block the oxygen supply by going through the small vessels, and by blocking the small vessels the capillary networks which are commonly created around cancer cells, so that cancer cells are supplied with nutrients. So you block those, that is number one. Number two is that you supply the medicine which is loaded with small beads to the target tumor cells. So now this entire process of course can be trialed with a human being, and then you can see that whether it is becoming successful or unsuccessful, or it can be trialed with an animal. You know that many times drugs are trialed first with animals and then with humans, and then drugs come to the market. Can we make a in vitro microfluidic device or an artificial microfluidic device which will mimic this network, and instead of making a trial with animals or human beings, we can make a lab based trial of whether this cancer treatment is working or not. So in reality how this drug works, I will try to run a movie. Just I would like to let you know that this movie is obtained from a commercial source, but we have no commercial interest in showing this particular movie. This movie I have obtained from one of my collaborators, Dr. Zhongli Zhang from the University of Southampton, and we work on projects related to this, and we have authored papers related to this, but having no commercial interest or conflict of interest with what I am showing. So just have a look into it as purely from academic or scientific point of view, and do not be bothered about any particular brand name or any particular company which is related to this.
Hepatocellular carcinoma, or HCC, is one of the most common cancers worldwide, and the third highest cause of cancer-related deaths. Less than a third of HCC patients are eligible for the curative treatments of resection, ablation, or transplantation. For these patients, the only treatment option with a proven survival benefit is transarterial chemoembolization, or TACE. DC bead is a drug delivery embolization system that brings a new level of accuracy and tolerability to the TACE procedure, precision TACE. DC bead is composed of a sulfonate-modified hydrogel macromer containing negatively charged sites. This allows DC bead to bind with positively charged drugs such as doxorubicin. Using aseptic technique, doxorubicin is loaded into DC bead. The positively charged doxorubicin binds preferentially to negatively charged sulfonate groups, displacing water from the bead during the loading period. Precision TACE is a minimally invasive catheter-based procedure. The drug-loaded beads are drawn up into a syringe and mixed with non-ionic contrast medium to aid delivery and visualization. Following angiography, the catheter is inserted into the common femoral artery and then carefully positioned in the feeding vessel of the tumor. DC bead is slowly injected into the catheter. The DC bead drug delivery embolization system has been specifically developed to contain the chemotherapeutic drug until it reaches the tumor, maximizing tumor delivery and minimizing systemic exposure. The calibrated nature of the beads ensures targeted vessel occlusion, stopping intratumoral blood flow and causing ischemia and tumor cell destruction. Once at the tumor site, DC bead starts a controlled release of the drug. Doxorubicin enters the ischemic cells, preventing DNA replication and destroying membrane proteins. This results in apoptosis and cell death. The controlled drug delivery from DC bead is sustained over an extended period of up to 14 days, with peak drug delivery 3 days after the precision TACE procedure. Tumor necrosis can be seen 2 to 3 weeks after delivery, resulting in reduced viable tumor burden. Due to the targeted nature of the drug release, virtually no drug-related toxicities have been reported in published clinical trials, and pharmacokinetic data show a 2-log reduction in systemic levels of doxorubicin compared to conventional TACE. The high tolerability of precision TACE should support patient acceptance and encourage adoption of this new and highly effective treatment option. DC bead delivering precision TACE is the new standard in safe and effective treatment of unresectable hepatomas.
So now, how do you make microfluidic channels which mimic these complex structures? So microchannel network design. So you can make microchannel network design on the basis of microvascular geometries and bifurcation structures and blood flow conditions of selected vascular regions. A range of bifurcation shapes can be designed using CAD software, and then devices are fabricated on 2 PMMA sheets using micromilling protocol. Alternatively, PDMS based microchannel can be fabricated by soft lithography. But the question is, are these channels, and it is a $1 million question, are these channels which we are trying to fabricate as flexible as that of blood vessels in human bodies? So that the idea is not just make complex networks of channels, but complex networks of channels which are biomimetic microfluidic devices. So one can cast typical materials, for example collagen, into a PDMS gasket with a needle held across the casting chamber. On collagen polymerization, the needle is extracted to create a hollow cylindrical channel with a collagen material. Collagen is related to tissue engineering. So it is a soft material which is a tissue material, and that kind of material if you use for making these microfluidic channels, and then as a next step, if you could possibly line this with endothelial cell lines, that is if you have endothelial cell depositions on these collagen based networks, then you could possibly make God gifted, God made channels by making microfluidic, by using microfluidic techniques. So if that is what is common, that is what is possible, then many mysteries in human body can actually be unveiled by microfluidic experiments done in laboratories, and that is where the futuristic direction in microfabrication research should go to. The job is very challenging because the flexibility of the microfluidic channels in human bodies, that depends sensitively on many things. One is that it varies sensitively with local blood pressure in a very unknown and in a very complex manner, and that is, it is very difficult to mimic that in a paradigm of microfabrication. So how do we make microfluidic channels which are as flexible as that of micro channels in human bodies? So if we can make that and if we can mimic that, then there are many mysteries in medical science and technology which can actually be resolved, which can actually be solved, and then one can make small, I mean one can then mimic small, I mean collection of cells which is tissues on a microfluidic chip. One can then go for a collection of tissues to make organs on a chip, and there are concepts of organs on a chip, but the organs on a chip, the concept, from concept to the realization of organ on a chip where it will behave functionally exactly like organs is something which is a matter of great importance. So and then if you go further ahead with that, one can go ahead with a collection of organs on a chip where the microfluidic networks mimic, microfluidic networks connecting the organs mimic the flexible networks, flexible channel networks within human bodies, and then that is going to give rise to the paradigm of human body on a chip. So one can see that starting from the very basics of microfabrication, how can we go ahead, and these are not just fancy stories or fairy tales. Research is currently going on in many laboratories in the world towards these directions, and if these are the real possibilities, and then one can really revolutionize the medical engineering and medical advancements in medical science using microfabrication. Thank you very much. We will stop here today.